on eukaryotes today. So I will give you an overview on some of the questions that people have been asking with fungi, and then end with some data on the built environment that's been looked at with regards to fungi. We're part of a group that's trying to develop the data analysis course. So a lot of what I'm going to talk about are tools as opposed to results per se. But I will just start out with to emphasize that you can tell when there's a problem in your house when there's a big mold, right, that's growing. This is Serpula, can cause rot inside buildings, and that's pretty obvious. But in fact, there are, of course, lots and lots of different fungi that live inside buildings that we don't necessarily see or come in contact with. And this is just an example of a whole bunch of different cultures that were collected, and you can see lots of different forms among those different fungi. So there's a lot of interesting things going on inside the building. Okay, so what I want to emphasize again is that fungi, since we haven't had a chance to show beautiful pictures of fungi yet in this meeting, I thought I would emphasize they're pretty. They make all kinds of beautiful fruiting bodies and different structures. This is lots of different scales of both microscopic and macroscopic forms. They put a lot of energy into reproducing and making fruiting bodies to spread those spores. And so I just want to emphasize that there's lots of interesting biology to go with these organisms as well as the bacteria. And that they're an old kingdom, and that, just to emphasize again, we with the advent of molecular phylogenetics, we were able to really establish the relationships among most of the groups much better than we could just with morphology, and it's important to use that backbone to study differences in changes in structures, sort of morphological structures or lifestyles. Most of the fungi that we'll be emphasizing here are going to come from the Ascomycota or the Basidiomycota groups of fungi, so just to kind of orient you phylogenetically. And again, fungi, we've heard several talks about fungi, about bacteria interacting with organisms, and I just want to emphasize again, fungi interact with other organisms too. They both interact in sort of, you could say, positive manners and mutualistic manners in terms of making mycorrhizae that associate with roots and are able to exchange nutrients, as well as lots of fungi that grow within plants, and in fact are a real hidden world in terms of diversity that are being described now. Betsy Arnold's lab at Arizona has been a pioneer in describing these different organisms, and you can see just an amazing array of different forms found within sometimes just a single leaf. You can find tens or dozens of different species. And they also, of course, can interact, what we might say, negatively. I'm just showing you some of the least gross pictures, but different kinds of dermatophyte type infections. Actually, my dog has an infection just like this and has nothing to do with me working on fungi. But so at any rate, they also do interact with those little bugs, those little bacteria bugs. And in fact, we do have examples of bacteria inhabiting hyphae of fungi and presumably interacting and producing some sort of communication and interactions. They, of course, are also really interesting symbioses with fungi and, for example, viruses, where you have a fungus plus a virus is necessary to cause a plant to be able to be thermotolerant. And so that you can do things where you cure the plant or the fungus, and it doesn't survive in heat. If you add the virus alone, it doesn't do it, but if you infect the fungus with the virus and put that back in, you're able to have protection and thermotolerance. So lots of interesting communication that goes on there. And of course, we also have insects and fungi that work together in terms of farming. So there's a really interesting symbiosis that's millions of years old in terms of ant-farmed fungi as well. So lots of interesting areas of research in terms of interactions. And in terms of how many fungi are there, again, with the advent of molecular sequencing and the ability to describe species in a culture-free way, there have been several different debates about how many species of fungi there are. And so Hawksworth presented a hypothesis that initially estimated the number of fungi to species to be 1.5 million, and this was based on this ratio of if you took a plant, again, not knowing about endophytes very well at the time, or not having a very good accurate number, saying that a given plant species might have six different fungi associated with it. But then as we started to sequence and look more deeply at things like soil or other insects, we realized it's a much broader much more diverse set, and so there's been recent estimates sort of in the order of 6 million species, but I really think that we still have a lot more ground to cover to be able to really get a good estimate on this, and in part that sort of informs our ability to look at these saturation curves of species counts and know whether or not we're really getting close. So in order to tackle some of this diversity, fungi have been one of the biggest groups of eukaryotes that have been sequenced. And so at least 400 genomes of fungi exist today, but I would say at least double that are in the pipelines or proposed through many of the sequencing centers. And so it's a really rich kingdom for which we have a lot of different kinds of sequencing data, and so the important aspect, of course, is to use that data and be able to ask questions of it. But the sampling that's been chosen has been focused in part on the funding. So a lot of the funding was focused on things that were pathogenic or groups of really industrially relevant fungi. And so the problem with that is that we lose a lot of the phylogenetic diversity and our ability to really study the whole kingdom in an evolutionary sense. And so to try to tackle that better, we've launched a project through the community sequencing program of the JGI to sequence 1,000 fungal genomes, to be able to sample two families from every, two genomes from every family of fungus. So we believe there's about 500 families within the kingdom, and so if we sort of do a Noah's Ark and take 2 species per family, we can cover the diversity. And so you can see in some cases we've done a good job and covered a lot of the diversity of the different families. They've been really well sampled, so some of the Sordaria mycetes and Tophragnomycetina have been sampled very well. Sorry, they have things that not very many things that are remaining. So this is the completeness on this right side. But we have lots of families where we have nothing has been sampled, and so we're really working hard to try to close those gaps. So again, having data is great, but being able to do something with that data is even better. And so one project that we've launched to be able to try to analyze this data and bring the tools to the desktop for the scientists who aren't really bioinformaticians is to build databases to be able to interact with that. And so we have a system that's built on the UPathDB system, which I'll describe in just a second, but it allows us to do data mining of genome data and be able to save those queries and share them and replay them. And so this is built off of the successful PlasmoDB, which has also launched a couple of other species for different species, MicroBEnet and AmoebaDB. And again, these are all focused on, typically have been focused on human pathogenic organisms, but the FungiDB is intended to kind of cover this whole kingdom. And so our latest release has 31 genomes in it, but we're sort of ramping up for a much larger release in the next 6 months. But this covers some of the phylogenetic diversity as well as some of the medically relevant fungi that have a lot of genomic resources. And so this system allows you, I'm just going to walk through very quickly what this system allows you to do, but you can search for things like all the genes in an organism. That's great. You can make a big list. So here's Cryptococcus neoformans and Cryptococcus gattii, and we can take through this query and get, for example, a list of all the genes in the genome. We've already pre-computed orthologs, so we have sort of this count of the number of genes in each species, and then we can see those broken down by ortholog groups. But what's really powerful about this system is that we don't want to just look at all the genes in the genome. We might want to start with doing an analysis. So here's RNA-seq data that's been loaded for a knockout strain, and we can, for example, look for all the genes that are twofold up-expressed in this knockout strain versus the wild-type strain. And so we get back a list of genes. That was easy. That was 2 clicks for the user. And then what's nice is, so we can look at this and say our model system H99, but we can also ask how many of those genes are actually found in another species, and so we can do these query combinations, and so we can intersect that query with something like all the orthologs. So we can take this other species of fungus and ask for the orthologs of the genes that came out of the first step when we get this transformation of the genes there. So the gene count number may not be 1-to-1 because some of these genes may be paralogs, but it allows us to sort of do this transformation of the data and, really importantly, transfer annotation and function information from a model system to perhaps a system that isn't as tractable. So we think this is an important tool for being able to ask questions about the fungi. And so in addition to understanding where the fungi are in the buildings, we want to know what they're doing. And so being able to look at these kinds of genome analyses on fungi that might be found indoors is going to be important too. But in terms of being able to ask questions about microbial ecology, the Sloan Foundation funded a data coordinating center that includes our lab as well as Rob Knight's lab, who's developed the CHIME tool, Mitch Sobin's lab, who's developed the VAMPS tool, and Volker Meyer's lab, which has developed MG-RAST. And the idea is for researchers to be able to upload and import data sets of sequencing of either fungi or bacteria and amplicons, and be able to analyze their abundances and do typical analyses. But I'll explain in part why this has been difficult with fungi. All of these tools were developed with bacteria and 16S in mind, and so to try to transform that towards fungi is a little bit more complicated. So one of the problems with fungi is what they're called, which is that in fact, many fungi have two names. Many fungi have more than two names, but many fungi have two names, which makes it very confusing to teach mycology, because you have to ask people to memorize two different names. But in part, it just becomes very confusing, and this has to do with the fact that fungi have sexual and asexual stages, and so oftentimes, without molecular data, they're identified separately and given separate names. Okay, well, with DNA sequencing, you can say that those two things are the same thing, and so you shouldn't be calling them two names. They really should have one name. And so this Amsterdam Declaration that was put together last year has said basically, we need to stop doing this. They all need to have one name, and you need to stop doing it now. So in fact, like next year, and now it's already sort of outlawed, and next year you get flogged if you do it, so you can't publish. So at any rate, they've had these different interesting sort of symposiums to be able to really debate this and get this out in there in the open. Okay, so how do we identify these two species are the same, these two organisms are the same? Well, we can use these marker sequences, and so again, this has been part of the debate, was what is the right marker sequence that's best able to discriminate. So we have the 18S gene as part of the rDNA loci, but that doesn't necessarily have the resolution to distinguish species, and so the internal transcribe space, or the ITS sequence, has been very successful. So you can see it kind of diagrammed here. And in fact, just this week, so Berkeley put out an announcement for this paper. This actually is a book chapter that was cited 10,000 times. Okay, and as the Berkeley press release said, Saul Perlmutter won the Nobel Prize, and their paper is only cited 8,000 times. So here we have this book chapter on identification of fungi that's been well cited, and again, it's because this was an important advance in the ability to distinguish these individual species. And so lots and lots of different primer sets have been developed, and people have optimized these for different groups of fungi or for sort of generalist primers. But this has kind of been the agreed upon marker. In fact, it's agreed upon because the group of people that were the most ornery and wanted to discuss this the most agreed that this will be the marker for now. So, and it's important that this debate happens so that we can agree on it and understand the arguments and move forward. And so again, just in the last year, this has been accepted as the way forward for identification. So all these herbaria will be typing their specimens. All new species that are described have to have this sequence deposited when it's named, and this is going to be an important way forward. The question is, how do we deal with this backlog of the 100,000 sequences that are in GenBank that may or may not have proper names? And so that's partly what our group is working to help happen with lots of collaborators, including many people that are part of this project. So that's what we're faced with. We're faced with kind of a messy database that we're trying to make more useful, but I will show you at least one analysis of an indoor environment just so you can get a sense of how this has been done. And this is an example of indoor air or indoor dust that was sampled across several different geographic locations. And they used the ITS region, and they also sequenced the LSU region. And they used 454 for this, and so that helps because this locus is pretty large, and so it's good to be able to get most of it. And so the main finding they found out of this was that in fact they found increasing species diversity with increasing latitude, which was not what they necessarily expected. And so if you plot the places, there were 72 sites sampled, and if you plot them on the globe essentially here, and you can sort of start in the middle at the equator and go out, you can see that there are many more dark boxes indicating that these species are found, and so you can see that there's many more dark boxes in the upper and lower latitudes, suggesting that indicating there's much more diversity within those groups, and that was surprising, because typically we see much more species abundance in the tropic regions, and so this may or may not be a general rule. This is probably the rule for these types of buildings at this point, but that is the observation from that sampling. And again, you can see some variation in the types of different major groups of fungi. If you're not an aficionado, these names don't mean much to you, but just in terms of there are differences in some of the different groupings that you can see in terms of the abundances. And again, trying to understand how much latitude plays a role seems to be one of the more the driving force in that kind of in that diversity. So we took this data again and analyzed it within the system that existed for the sort of fledgling system that existed for ITS data in our Sloan project, and so we used some tools that Volker Meyer's lab had developed, and if we just plot all those samples again, in fact, they did two different markers. They used the ITS marker, and they also used this 28S marker. And what you can see is that nicely the plots separate out very well, very similarly between the two different markers. So they seem to separate the samples quite well in these MDA plots. And then if you overlay other kinds of metadata on it, so for example, if you overlay country, you can see again that you have by country, they seem to be grouping fairly well by some of these colors. And you can see again that the two markers behave fairly identically for that. And if you look again at something like elevation, again you see pretty similar groupings there as well. So I think this just sort of shows some robustness in the markers for this kind of identification, which was helpful, but also that sort of our simple automated methods were able to replicate what was done in that more complicated analysis in the paper. So the thing is, again, identifying lots of those species, so that the challenge is many of those things are just barcodes. You have no idea what those are. We put genus names on there because that's the best guess from a similarity search. But in order to actually take those organisms and try to figure out what are these things that are living inside the building look like, are they different really from the things that have been routinely isolated from soil or from the environment? And so what Keith Seifert's lab is doing through a Sloan-funded grant is actually doing the serial dilution and able to essentially isolate down to the single cells from these dust. And I didn't put the whole series of pictures that he has of the pile of dog hair in his house, and then the breaking that down into sort of liquid culture and then being able to actually isolate it. But you can imagine the pre-steps. But at any rate, he's able to then go through and actually take these global dust samples and actually go down to the level of individual organisms. And so this is really important for trying to understand whether or not the things that are living inside really are different than what's outside, or are we seeing whether or not these barcode sequences really are able to distinguish them. So that's part of what us also going on to try to follow up on that. But it's not particularly high throughput, but it is the way forward on some of those questions. So just wanted to give you a taste on some of the tool development that's going on. It's sort of a preview for where we're going to go with some of these ability to work with ITS sequences to be able to analyze these community data. And the important thing will be we're having a Sloan workshop in a couple of weeks that will help us actually have communication among the different groups that are building databases for ITS to centralize this and be able to communicate and be able to curate is the important thing, is to have this feedback once you figure out what something is. From a barcode, many of these are unknown sequences that we find. Being able to turn those into something known is going to be the next step in making that resource useful. There's lots of different folks that work on these projects from the different labs that are all funded through the Sloan project. The FungiDB project is a collaboration with the groups at University of Pennsylvania and University of Georgia, and there's some programmers in my group that have worked on that, and then Anthony and Keith, who've were nice enough to share a lot of their data before it was published, and the two funders for the FungiDB project. Here, and this picture is taken from what we call a mountain, although it's a very small mountain, Rubidoux, looking up at where we are sitting here in Lake Arrowhead, so sort of the reverse view from here. So, okay, with that, I'll be happy to take any questions if there's still time. Thank you. Yeah, we have a couple minutes. Okay. Yeah.
So one thing that comes up that we sort of thought with, you can use the ITS primers to see who's there and get a count of the relative amounts of different kinds of stuff. The absolute amount is nice to get too if you can with qPCR or something. Have you tried to adapt these same primers to qPCR for absolute amounts, or could you comment on that?
Yeah, I think, there's a couple of challenges with that, which was mentioned in Jonathan's talk about copy number. So a lot of copy number variation among species, and I really don't think that we have a handle on it, because in part, even, the genomes aren't really assembled necessarily with the ITS region assembled perfectly. So I don't think we have a baseline to count from that. I do know a few people have done quantifications with qPCR with those primers, so I think it has worked. I don't know that it's been widespread. I'd have to go back and see the literature for that. But I think, yeah, you're right. It's a very important point to say how much of everything is there, not just what's the most abundant.
I think the length variation, though, for the ITS is hard for qPCR. We have an 18S primer probe set that's coming out in BMC.
So, yeah, so maybe not using ITS, but using 18S is going to be more accurate for that. Or unless you have, maybe really primers for a very specific group, that might help too. Yeah.
So for the latitudinal gradient stuff, I'm just curious about how similar the environments were you're sampling. Because if you're sampling estuaries versus something less diverse, that'll have a big impact.
Sure. And again, this was Anthony and Tom Bruns and Keith's study, but their sampling was focused on office buildings, as far as I know. And so it was they did try to. They were focused on buildings where people worked. I don't think there were any kind of. If they were residential buildings, I don't know that they were not like air conditioned, because they were using vents, I think, or using filters for most of the places where they were collecting the dust. As far as I remember, maybe we could double check from the methods again from that paper, but I think they tried to control for not having hugely variable types of buildings, so that wouldn't contribute as much. Yeah.
I have a follow-up to this. The amount of lignin that exists in wood in the area is going to increase as you go to higher latitudes.
Yeah.
Does that have a difference in the population, therefore the results you see?
Yeah. So the increase in lignin of the environment where you're sampling from is definitely going to increase as you go into the sort of more deciduous forest, and so you would see a real difference in presumably the communities. I don't know if they necessarily tried to control or tried to ask that question to see if the species that they were finding really correlated well with those that would be best associated with the higher lignin environment. I'm not sure, but that would be another explanation for it. Yep. Okay, thanks very much.
Move on?
Okay.